Hello, everyone. Taste perception starts on the tongue and soft palate, where specific chemicals in foods and drinks interact with taste receptors found on the taste receptor cell. Now, taste receptor cells are grouped together to form taste buds across the surface of the tongue. And each cell within the taste bud has the ability of responding to one of the 5 elemental tastes, namely sweet, sour, salty, bitter, and umami. Now, here's an enlarged picture of the taste bud on the left-hand side of the slide. And on the right, we have an example of a bitter tasting chemical interacting with the bitter taste receptor, which is on the surface of the taste receptor cell. And the taste receptor cell forms a synapse with a nerve that sends electrical signals to the brain where bitter taste is perceived. And now for some audience participation. I believe that you've all received an envelope. If you'll open that envelope and taste what's inside, we can determine your level of sensitivity to the bitter compound PTC. Okay, so go ahead. I love these expressions. That's the strongest one that I saw. No, that one. Yeah. That's very strong. Yeah. Oh, man. It's not that bad. It's PTC, phenylthiocarbamide. Really? You don't taste it? Okay. All right. By show of hands, does anyone taste this chemical? Who can taste it? Okay. Yeah, okay. Who does not taste it? Yeah, okay.
I would estimate that roughly 75% of you were able to test. It was almost like classic, like 25% couldn't and 75% could. And so one thing to think about, remember there are 2 common variants, right? And think about the different combinations you can have. So you could have 2 of the, taster and be heterozygote or not. And one thing we should mention is that the taster haplotype is dominant. Yeah. So think about that and think about why you would get a 75 to 25 ratio.
Okay. The ability to detect PTC bitterness is often cited as a classic example of a highly variable trait in humans. Individuals who find PTC bitter are classified as tasters, and those who find PTC tasteless are considered non-tasters. Now, the ability to taste PTC is also correlated with the ability to taste naturally bitter substances in food. PTC has been used in taste perception studies to understand the link between genetics and taste perception. Now, dietary sources of bitter substances include vegetables such as broccoli, bok choy, cauliflower, cabbage, watercress, etc. And these foods contain bitter-tasting phytochemicals, as well as antioxidants, fiber, vitamins, and minerals. Although bitter substances are present in beneficial foods, many toxins in plants are also bitter, so it's been hypothesized that bitter taste perception likely evolved in humans to prevent the consumption of potentially toxic chemicals and may represent a defense mechanism in our species. Now, the bitter taste receptor gene TAS2R38 on chromosome 7 was discovered in 2003, and this gene accounts for a large proportion of the phenotypic variants in PTC perception. This gene consists of 2 untranslated regions depicted here in blue and 1002 base pair coding region depicted here in red, which encodes for a 333 amino acid receptor protein product. 3 SNPs within the coding region of the TAS2R38 gene have been found to explain up to 85% of the phenotypic variance in PTC sensitivity. Now, these polymorphisms can cause amino acid substitutions in the 333 amino acid receptor protein. For example, variants at nucleotide position 145 encodes for either a proline or an alanine, and those are 2 amino acids. Variants at nucleotide position 785 encode for either a alanine or valine, and variants at nucleotide position 886 encode for either a valine or isoleucine. Now, the different combinations of these amino acids in the receptor protein can give rise to different amino acid haplotypes. For example, when proline, alanine, and valine occur together in the same receptor protein, this particular combination is known as a PAV haplotype.